Hello?
Good morning.
Morning. How are you?
Okay. Been
Good.
a interesting couple weeks, really, but I'm alright.
Good. That's good. I'm glad to hear you're doing okay. I know it's been pretty busy. You've had um, your fellowship that you've been kind of working on, and um, we've been, yeah, fall term is going by. So we've got actually, we submitted the abstracts for Neuromatch this week, so, and it looks like they're accepting, they're kind of like dragging along the deadline. Oh, hello, Angela. They're dragging along the deadline, so now people who want to do flash talks are, have a deadline of like, I don't know, the middle of the month. So I think they're still accepting flash talks. And
That's good because I really wanted to. I feel kind of bad. I sent you. I don't know if you got the email from Rebecca or involving Rebecca last night, but I was going to. I connected with her again last night after mentioning a couple weeks ago,
yeah.
and I used the wrong email when
Oh,
I did that. Apparently, there was two G's when it was supposed to be one G
oh.
in some things. So that was a. I feel kind of bad because I really wanted her to join for that. So that she can still
Yeah.
join up Neuromatch, and I don't know if she's looking allowed or not, but we'll see.
Did she, what do you mean? Like she wanted to submit something or she wanted to.
She was interested in Neuromatch and interested in working with the lab, kind of on the VR project.
Okay.
Um, So I don't. I think it would be. I think it'd be great if she just goes to Neuromatch, and you know, um, I think that'd be a good step for her. Like if she ends up doing something with us specifically, that's great. But we'll see.
Maybe I'll contact her and inquire. I mean, I didn't want to make her think that we weren't interested.
No, and I felt bad because I interpreted her lack of responding as not being interested, but
Oh, right,
also
right. It was
I
a.
it was the wrong email too. So
Okay. Yeah. Well, that's good. I was going to respond, and then I said, "Well, I wanted to." I saw that you had responded, so maybe I'll reach out and say, hey, we're still here, and definitely, it's difficult for people to get over that hump. Like sometimes you have to encourage them. Sometimes it's, you know, they don't really know what they want to do, and then you have to kind of
yes.
give them like maybe a little bit more distinct options, like I want to work on VR. You
Yeah,
know,
and Rebecca's someone who is doing like neuroscience and psychology stuff. And wants I think she wants to do something. She wants to get to graduate school and do something in psychology. I forget what. So it would be a really good opportunity for her, but it's not quite. She's still getting used to doing those kind of things. So yeah.
yeah, it's always a thing. Like it's always a hard thing to sort of get into. I guess once you get an established pattern, then it's a lot easier to make time for it and to figure out what you want to do. And but that's good. Yeah, I'll reach out there. I'll see what she's interested in. Maybe a bit more, just a bit more detail about that. Um, good. So it looks like Morgan's here, and so does anyone have any updates before we start? And Angela was here too, but I don't know. He's probably reconnecting.
I mean, my major update is probably going to be Neuromatch and grad fellowship stuff related things. But I don't know if we want to talk about that more later.
Well, yeah, we can talk about that a little bit later. I guess I'll jump right in. There she is. I guess I'll jump into the Slack review. So we've had, again, a lot of activity on the different Slack channels. And so let's go into that. And again, if you have some comments, please jump in and we'll talk about them. So the first channel is Brain Organoids. I'm just going alphabetically. You can see my screen.
It looks just dark to me, but it might be me.
Let me reshare my screen.
I can see it.
Okay, good. Thank you. I will do that anyways, though.
I have to log back in, actually, so we'll see.
All right.
I'll be right back, yeah.
All right. So the first channel is Brain Organoids, and so we have, again, a lot of papers. Thank you, Morgan, for posting things in the channel. There was a crowdcast on biomedical and translational biomedical engineering. There was this Google Groups Brain Organoid group, and I couldn't really get into that. I clicked on the link and tried to join. I don't know what happened, but
same problem.
Oh yeah, I don't know what happened there, but and then the Tarasky talk. So this is the Tarasky Institute. This is viscoelasticity from stem cell fate to new medical devices. So that's another. I don't know if this is recorded or not, but
yeah, I'm gonna check if we can get archives of these because they've got an incredible speaker series.
Yeah, it looks interesting. Then there's this challenges of determining how neurons are connected when you can't visualize the connections. This is a Medium post. So this is a paper from their group.
And this is somebody who works down in San Diego, has done work together with Alison Moultrie, the head of the UCSD Stem Cell Center.
Yeah.
And is right along the lines of orthogonal research and education lab in terms of application of computational mathematical techniques to biological systems.
Yeah, let's take a look at the article here. So this is a peer-reviewed paper published by their group, and that is Frontiers article. Super selective reconstruction of causal and direct connectivity with application of in vitro induced pluripotent stem cell neuronal networks. So this is really interesting. So this is taking, so induced pluripotent cells are things that you grow in a dish and you can convert them from somatic cells that you find in your body to stem cells. And they're pluripotent, so they have a limited number of fates, but they can move to different fates. And then you're able to use those because they represent someone's genetic background to, you can create neurons, and those are like sort of personalized to that person who gave the cells. So the donor cells are the ones that you're looking at, not just some generic genome. And so now they want to create neurons, apparently, and then they want to sort of map out a network of neurons. So, yeah, this is interesting. Actually, I think they're actually trying to do this with induced pluripotent cells. So they're not actually creating neurons per se, but they're actually looking at.
I think it's a more general problem.
Yeah. Yeah.
And it's good. I'm making a note for myself that it's good to remember that inducing pluripotent cells into neurons is still different than grabbing neurons from the subject nasally.
Yeah.
And using those, which is you can do, and you can do that as an outpatient procedure. So
right. Yeah, the way they usually do it is there's, you would go like to an induced pluripotent cell. Yeah. You could do that. You can get neural precursors from different parts of the body, but then there's also the route of going to an iPSC, and then you do another transformation where you take, I think 3 factors, depending on what neuron you want, and you put them into the cell, and you can get neurons that are sort of generic neurons. It's kind of hard to say. This is where you get into like, how do you identify a cell? Do you identify a neuron as like, is it a motor neuron? Is it some generic neuron? And that's the kind of the thing that, you have to work on there. But yeah, this looks like an interesting paper. It almost reminds me of the embryo network stuff that we've been doing in the other group, where we've been looking at in the embryo, there are these networks. They're sometimes gap junction related networks. Sometimes they're spatially distinct networks. So it looks like they're doing something similar. They're building, and I think I saw another paper where they did this with stem cells, where they looked at sort of they used network theory to sort of classify colonies of stem cells or induced stem cells. So this is something that you can do. So they're basically looking.
Yeah. This is
at the network itself. They have correlation triangles. So the idea is that you would have, like, if you have a network, you have these elements in the network, like, of connectivity. So you have like a triangle where 3 nodes are connected together, and then that is something you can classify. I don't know if people caught the crowdcast yesterday, but there was they talked about, they were actually, this topic had come up. I know Morgan was there. I saw him in the chat, where they had this thing about, they were talking about network theory, and they were talking about how...
Oh,
yeah.
You're talking about the learning salon?
Yeah, the learning salon.
Yeah.
Where they said, well, it's great. A nice global measure of what's going on in the brain, but like locally, it's not necessarily a very good measure, which is true. It's like network theory is not, it's supposed to capture the global state of what's going on in the brain. So you build a network from all these neurons, you say something about it like they're clusters of neurons that are highly connected, and then there's, what else can you say? Can you make causal relationships or not? Yeah. Probably not. But when you go down to the level of what they call motifs, which are these patterns, I think this is the paper, these patterns like correlation triangles and things like this, or sometimes they're just kind of like stars or, different shapes that form in the network, then that's something that's more local, and that can maybe tell you something about the relationship between those specific neurons. So... And
yeah, I mean, so I just wanted to say, in relation to the learning salon, that, I think it was John's criticism was, I think, pretty spot on in this, as a neuroimager, it was very spot on in the sense that, like, I've only seen us use graph theory to, in this kind of class identification way.
Right.
In terms of healthy controls, some sort of patient population. We don't really know what we're getting. We're just getting a difference on this metric.
Yeah.
And it was. I felt attacked, but in a good way.
Yeah, I mean yeah. So that's something that is, always with methods you have, like, you can't broadly apply them. Sometimes they're very specific to something, but
yeah. I mean, just somewhat related to that, though. I just want to flag this. I didn't drop it in. This new Steve, is it Strogatz?
Yeah.
Steve Strogat's paper with a researcher from Santa Fe Institute that might say more about the kind of temporal dynamics that will occur on graphs. Like that would be useful if we identified these kind of like 2 graphs, and then we knew that the temporal dynamics would differ in some way. I think that would be a lot more informative and would kind of speak to John's point about, like, what's this mean except like identifying, patients.
Yeah.
We could say something about like, well, what that means that on this kind of graph, you're going to see less synchronization or something like that.
Right.
I'll find the link.
Okay, yeah, please do.
Sorry.
So there's this link to Conception Bio, and this is something I'm going to talk about later. So we'll skip over that for now, and I'll come back to that. Data Science ML is another channel. I wanted to go to that because we've just been talking about networks. So this was last Saturday. We talked about some interpolation of machine learning, I believe. So that's some of the links there. And then this is something that Ankit posted in the chat here. This was the demo that he did on GA reversals about 2 weeks ago, where he showed this genetic algorithm that he would reverse the parts of the algorithm, and then they would produce this plot. So, Ankit, you're here. Could you explain this plot to us? Okay. Could you explain this plot to us a little bit? Are you there, Ankit?
Excuse me. That does look odd for a... Sawtooth doesn't...
Oh, yeah, these are the reversals here. So you've got these. So this is the objective score. It drops, and then it comes down to this. So it's kind of like, it's converging here somewhat, and then it's going back up to its value. So I was just kind of hoping that he could explain that, or, what the... Okay. Well, we can come back to that if you want. Anyways, that's his work that he's doing he's refining this algorithm, and there was a bug in it in the original demo. So this is the reversals that you're getting. Basically, you're getting these reversals. You're switching the algorithm, and it's kind of settling back down to where it's converged. So it's converged here, and then it kind of goes up and then down. And the idea would be that you would have that it could theoretically improve performance as you're switching. It doesn't show a lot here, but like there's this would be the idea, especially on a rough problem domain. If you had something where it was a very rough fitness landscape for the algorithm, this would be something that would help it minimize its objective score or cost function, or increase its fitness. So the next thing I want to I'll go to maybe the general channel. And general is, we have a couple things in here. This was from Tuesday. Ankit posted something about Gitbook, which is a tool it's not GitHub, it's Gitbook. And it's using the Git platform, but to publish different, layouts and things like that. So it's like a book. And Jesse said he wanted to look into that. I don't, I've looked at Gitbook a little bit. It might be useful to put out some of our things that we're pushing out on Notion and like aggregating a lot of those things. Because at one time we were just using GitHub and GitHub teams and things like that to organize some of these things, which is, it's adequate. But did you want to say something?
It looks kind of like it's... I mean, you can basically use basically the exact same... You can basically just use Markdown for Gitbooks, but the presentation might be a little bit more user-friendly than a GitHub page. And a little bit less cumbersome than making a GitHub-based site like in Jekyll or whatever. So I really haven't done anything with it, but that looks to be kind of the advantage of it.
Yeah, I think that's true. It's like the GitHub page. It's kind of like it's not. Well, I mean, I don't know. It's probably a little bit more user friendly. We'll have to check it out. I don't know if that's something that is, we always try out these tools and then sometimes they happen and sometimes they don't. So, well, anyways, that's one option for doing, like, stitching a lot of the stuff together. Then there's this Stanford, the human the HAI group. Yeah. The Human AI group. So they're hosting a fall conference on policy and AI. So this is sort of ethics, tech, and society oriented. And then this is the site, which is another event on ethical use of AI.
Yeah, I mentioned that to. I actually mentioned that to Valeria, one of our sort of alum from the ethics group. I think she's interested in that. But that's like a graduate school level training for people who are really interested in kind of tech and policy. It's a summer school, basically.
Yeah.
It's a summer course. I'm not sure it would totally fit me. I may apply to it, but there are some people, potentially Angela, and even some other people. I'm going to actually, this is glad to see it here because I'm going to re-mention it to some of the people in, we still have some strong ties to the UK effective altruism group.
Right.
Location. Not just because the people we were working on for Neuromatch with the Avery submission were mostly from there. But in general, I'm still involved in that, and we're all in like a Discord. I'm an alumni of that at this point, but there's an interesting, I don't really know pipeline, but just connection to that group still, and some there will probably be interested in that too. So.
All right, that sounds good. Then there's this open worm restoring on Hacker News, and
sorry, that was more a comment than.
Oh yeah. I looked it up.
But I go ahead. Yeah, I was wondering why.
Oh yeah, I looked it up, and it's this, and I covered this in the DevaWorm meeting a couple weeks ago. There is this article by, they're the Singularity people love OpenWorm because it's you can simulate an organism. Eventually, you'll be able to simulate a brain, and then you'll be able to download a brain. That's the idea, and it's kind of not like really what OpenWorm is about. I mean, it's just their idea. And then this article that was published by someone was like, this is the 10th anniversary of OpenWorm. So in 2011, OpenWorm started, and they started to really kind of publish their first software.
Yeah,
and they were like.
I was just going to say, this is like, it's like when people say, Neuralink's technology is going to allow us to download a mind.
Right.
And so it's like, that's not what they're working on.
Yeah. But anyways, this article was like criticizing, OpenWorm. They said, well, in 10 years, they haven't really done what they, it's kind of failed. And which, I mean, it's not, I don't know what the objective is. Some people are projecting objectives onto the project and then saying it failed. So I don't know. But anyways, that was the article. Yep. And then it's been passed around Hacker News, and which is, it's, I don't know.
When I looked at the comments, they were kind of what I expected. And, people going back and forth between, can you do this? And like, yeah, there are people, this project isn't necessarily doing it, but like you could add in all kinds of, cellular behaviors and things like that are much more akin to the projects like that are built on top of systems biology markup language and, biomodels and things like that, right?
Right.
Anyway, the only other thing I was going to say was that Stanford obviously wants you to pronounce it Hal.
Hal?
I know it's H. Like it's Hal.
Oh, okay.
I mean, I don't know if they say it but it's Hal. Right.
I'm so confused by that because people, and many of the things I said, they say human-centered AI. Well, it looks like Hal, and it's like. Is what's the L? And it's like, is this? It's just
maybe Is it like for copyright reasons they can't say how or something?
Yeah, they want to put like, any numbers next to it. It would be even worse. So it's like, all right, it's just
Wasn't Hal like a sociopath though? I mean, like, yeah.
Nice guy.
So Hal was given conflicting orders.
Oh, okay.
Yeah. I don't want to badmouth sociopaths.
Right. Anyways, okay. So, Ankit said about Gitbook. It's usually made for documenting stuff. So yeah, like documentation, and that's kind of what we're doing with the Orthogonal Lab GitHub. Some of the stuff we're doing in GitHub Teams are trying to do is document things like projects and things like that. So that might actually be a good option. There's always this balance between sort of like moving things to a new platform and then like saying that what we have is good enough. Yeah.
And I as a sort of like, to put on my pure lab manager hat for a sense, I think also one of the things we're at right now is like we're kind of at the, just to give context for newer people, we're kind of at the end of a series of specific projects, and in the verge of like transitioning to other specific projects. And so because of that, some of the tool usage is a little bit here and there. And what I would say, if anybody, and welcome Divyesh and Ankit's here too, and I think Angela was here, and I don't think Rebecca will be here today. But like, there's a lot of opportunity to say, I would like to form a project around this, and don't if just to kind of mention this, like, if you want to do a project on anything and potentially recruit other people in the lab or outside the lab to work on that, let's talk about the project, and then we can fit our tool usage to that project. Don't feel like you have to use GitHub to do this or that, or use Notion or whatever. So just to kind of mention from that angle, like, we can tailor the product. We can tailor. The most important thing is like making a project and a set of outcomes and what you want to do, and like going in that direction. And I was actually previously very, very focused on kind of moving to that demo or die or deploy or die direction, but I've kind of been, my major thing has shifted from a really busy summer of UK and abroad and the EA stuff that I mentioned and other things, and a new job and a startup, and now I'm in like graduate fellowship land and grad school stuff. So my projects my main emphasis, what I talk about in the lab, hasn't necessarily been, here is my project in a specific context that I'm pushing forward. But I also I'm saying that to represent, like, that is a totally valid way to do that. And I know the lab meetings aren't really structured on that right now, but they have been in the past, and they could be, especially something that you want to do. So if you ever want to develop a project, that's always on the table, and I encourage that, because that will shape things. So lab manager hat says that.
Okay. Yeah, I mean, and when you say project, you mean like it could be a paper, it could be like an entire topic. Like, Jesse has developed his interest in like cognition futures, and that's a project which has a lot of tendrils everywhere. So like they're papers, presentations, but they're also like papers that we're doing, and we're kind of closing out those things.
Yes.
And then starting new ones. So it's like there are different, like, projects at different levels of specificity. It could be like something like what Ankit's doing, working on a single paper or idea, and then maybe at some point that becomes maybe a project where we give it a name, and it can go like that. And so I don't want to restrict people or make people think that like this is unattainable because you have to have this project and you have to kind of like work out a mission statement, and it's much more flexible than that.
Yeah, and I was using the term project like very loosely. I don't mean like a big
Right.
research agenda for sure. Like if there's like, oh, I would like to do more research in this area, that might turn into a paper or a deliverable or a set of presentations. Yeah, that's fine. And I just kind of wanted to give context of like, I know we maybe haven't, our meetings as they've been seen here might not look like that's something that has been happening a lot, but it is. We're just kind of in a cycle of like a lot of the papers that we're doing for neuromatch and stuff have been look kind of at like the later end of those quote unquote projects or those lines of development. So that's why there hasn't been like a big in the past, like there's been like, hey, we're gonna start a brand new paper with a brand new whatever, and it hasn't really. It's not that we don't do that. It's just that we're kind of collectively the lab is in like a cycle of that right now. So just more about context and in that sense.
Yeah. Ankit said to Jesse, you might want to adjust your mic volume. I don't know, it's a little bit, but you can hear the not feedback, but like the maybe a little too sensitive. Yeah.
Stuck in something there, but yeah.
All right. Yeah. So let's go back to the Slack. So we'll come back to this because I know that Morgan asked about like, how to proceed on the organoid stuff. So we'll actually come back to that in a little bit. Okay, Divyesh said yeah, it gives a thumbs up. So next thing I want to talk about is ethics, society, and tech. And we have okay. So this is this is the thing of the week here. So this week there were a bunch of articles released with Facebook's name on them that were not good, and it's this sort of, I don't want to say it's well, there's a whistleblower that's been telling us how Facebook has been gaming their product to encourage antisocial behavior. So like
engagement.
Engagement, right? That's what they call it. But anyways, there are a bunch of articles that were published this week, and I didn't know if Jesse wanted to talk about that, if he wanted to go over some of those, or if you wanted me to do that, just very briefly.
I don't really have a lot to say other than the basic, Facebook's the kind of one of the a broader vision of what's happening is for a long time. This has been Facebook's agenda for a very long time. This isn't a new switch for them, but also the timing is kind of in. I don't know, and you've seen it in the articles or if it's common knowledge by now, but like, why are they pulling the trigger now? And is it simply because of the pressure that's upon them, or is it just like this was kind of they had an internal arc of when they wanted to do it, and it happens to be the end of 2021 anyway. But there's a lot of scrutiny they're under, rightfully so, and that we've talked about and covered in the lab before, and we mentioned the ethics, some of our team projects too. But right now, they're quote unquote rebranding as Meta, and I guess they're trying to be this gateway into the metaverse, and there's a lot more to say than that, but
yeah.
Did anybody catch John Carmack's talk?
No, didn't catch that.
Yeah, he's usually a kind of breath of fresh air. He doesn't speak in marketing terms.
Right.
And so he talked a little bit about the internal battles that they have with open source, but also just that in his opinion, he's not sure you can build the metaverse. It has to happen, which it would happen organically.
Right.
But I just wanted to say that related to this, because I think it's important that Facebook optimizes engagement, and Twitter's team, right, I think, to get ahead of things, said that their algorithms do the same thing.
Yeah.
And they and they're, they were trying to get ahead of it and say, like, our algorithms do the same thing, and we don't know why.
That's
yeah.
I'm not saying that Facebook is singularly culpable in this, actually, because the idea is that you want to drive engagement, so you drive around, basically you have people coming from all over the world at once, and they're interacting with each other, and they're not going to agree on a lot. Right.
Dismantling your civics integrity unit while there's an ongoing political battle. Not the same thing.
So there are decisions that you can make at the corporate level that are proactive or maybe reactionary, and that's kind of the problem. So, you're basically, you could think of it like, I apply an algorithm to a social problem, and obviously, you can make it better or worse. It's not like, but you have to have some awareness of the problem or the social context to begin with and say, this is, how this algorithm might affect it. And then, there's all this, we have like algorithmic auditing, which basically is a way to like know what your algorithm is doing. And then there's also, just kind of knowing what your algorithm is doing, period. Like, having a transparent sort of process of how it's engaging with data. And so those are all things that are, they can be applied in different ways. Sometimes, companies can be less responsible, sometimes more. But I want to go back to the Slack and just kind of look over some of the things we have. So we have the interview. So then after that was Monday. And then I think Thursday or Friday, Mark Zuckerberg announced that Facebook was going to become Meta and that they're engaged at the Metaverse. And of course, they're doing this stuff with VR, with Oculus and some other things. And so, I don't know. My feeling about this is that people have done VR before. And like, VR is one of those things that like there's a very deep history of hype surrounding VR. So, when people do VR, it's usually like, it's like the open worm thing we were talking about. You get people who have a certain expectation of it. That it, even back in like the 90s, people would kind of view VR as like this really super immersive experience, and, you could get lost in a VR world. And the VR of the 90s was not that. It was like, really clunky, and it was amazing to people because they had seen nothing like it before, but it was like not what was portrayed in the popular culture. So that's one thing that you have to keep in mind. The other thing is that, I think that Carmack is right, that this is going to be like an organic process that, if these virtual communities are going to form, and of course, social media is a kind of virtual community, they're going to have to form organically. You can't just create like this, framework and then say, well, this is what we're going to have now. And then, there's the whole story of Second Life, where they built this world where people could build their own communities. And I guess Second Life is still around. It's just kind of like that should be an instructive lesson to people who want to do this. So
it'd also be really interesting because Facebook, as I'm sure everybody knows, basically is not really, it's not really the place to be for the younger generations.
Right.
And like, to be discourteous, like a bunch of boomers arguing on stuff on Facebook, that's cool, but like, are they going to want to dive into the metaverse and populate it and talk that way? I haven't heard a lot about what their actual like plan and vision is for transitioning Facebook, or have there been any specific remarks about what has going to happen to quote unquote Facebook? And obviously Facebook is Instagram and themselves, which is where yes, I've never met a young person today. I don't ask, what are you doing on Facebook? It says, give me your Instagram, and that kind of thing, or like even TikTok. That's in the story.
Yeah. I have teenagers.
Yeah.
So I get told, Like nobody uses that dead.
Yeah.
Yeah.
And so, yeah, so
But
this, so this is,
I mean, the meta announcement seemed a lot like Alphabet. You know, honestly,
right. Yeah, I don't know what
it wasn't. Like maybe they want to try and make it a rebranding thing, but it's like we've gotten big enough that we need a holding company name.
Yeah. Well, anyways, this is the link here to like this person, David Pierce, has published like a list of all the stories that were published this week. So this is useful maybe for, and this is the paper here, or this is the list. This is like a curation of all the stories that they published, and this is useful if you're interested in like following up on some of this for the project. So there was a whistleblower who kind of exposed what was going on in the company, that they weren't being accountable, that they were actually engaged, they were not putting any sort of safety protocols up for some of the things that were happening. And so this is but these are the stories that are just breaking in the news. So Facebook's hiring crisis, engineers are turning down offers, internal docs show. Facebook wrestles with the features it used to define social networking. I mean, some of this stuff is like, like this is not really directly related to the ethics, but people are saying, look, I don't want to work for a company that's, has a bad, or is dealing with this so poorly.
The story that I think always gets lost here is the story of Facebook's content auditors.
Okay.
So these are the humans that have to step through the flagged content. Super important psychologically what's happening to these people, right?
Right.
And it is also it's an extreme case of what is happening to some degree to people who are getting exposure to this algorithmic escalation,
Right.
But I just want to, definitely have that. These are the lost or the, they're not even employees, right? These are they're purposely contract workers, because we can't deal with the health effects of that particular worker related injury.
Right. So yeah, that's a good story, and I don't know. These are all kind of like aside from this, so just to give you an idea of what's breaking here. So I mean, there are a lot of, like in India or in Poland, you see examples of where this is like inserting itself into the political process, and the social sort of discord and things like that. And there's this theme of choosing growth over safety. So, do you choose to grow your user base, or do you choose profits, or do you choose these safeguards and, making them better? And, so then there's also this formula for sort of driving different emotions or emotional states. And this is something actually that, this week we've been talking about allostasis and emotion. And I know Jesse, the EI group in, or what is the group in England? What's the acronym that they use?
Colloquially, it's known as like the EA Hotel.
EA Hotel.
Like, it's like they're trying to change their name because it's a bit awkward, but it's officially the Center for Enabling Effective Altruism Learning and Research. So it's sometimes called CELR.
Oh.
But like it's very clunky and
yeah, come stay
like
in the
EA
cellar
community in the UK or whatever. So I don't know.
Well, anyways, yeah. So that group, and I was thinking about this, and we were doing this abstract, is that they have this system, and Twitter kind of has this as well, where you assign points for different things. So like in this case, it was 5 points for anger, 1 point for a like. And so, this is their internal formula. If you go to the interface, you'll find that, they're like, you can do things like copy your group, maybe like in Twitter terms, it would be a retweet, or you would have a like, or, and then those things, people interpret those as like reflective of the content. And so the idea would be that, you have these things happening in the interface, and then it's driving people's emotional state. Or, the story, if they read it, it drives their emotional state, and they contribute to the system here. And so, there are a lot of interesting regulatory things going on with, social media content and emotional state, and critically that regulation. So, like, if I send a piece of content out into the world and it gets, 10,000 likes, that's great. I feel good about it. Maybe I shouldn't. I don't know, but it does. And then, if I get a lot of anger, that's also something that regulates not only my social state or my emotional state, but other people's emotional states as well. It's just something I thought about this week when we were doing this abstract. So,
I just wanted to.
Yeah.
It seems good to add to this. Patagonia has a thing touting their certification as a benefit corporation. That all these criticisms of Facebook's behavior seem to miss the fact that like there's no guardrails for Facebook as a corporation in this regard. I'm not sure what the guardrails are for Facebook in this regard.
Yeah.
Like, so yeah, just want to say that.
Yeah.
I'll put a link. I'll put a link in for the BLAB thing.
Oh yeah, that'd be great.
I think that's like that specific question is actually I kind of wish I could call upon Valeria right now, because she's from Chile, and so Chile just thinks a little bit differently in terms of how they structure their education, but sort of the equivalent to her thesis would be on accountability and technology and AI in this sense. She's a law student, and what are the guardrails for Facebook? Like, and how is there anything like it's sort of like a really interesting complex mix, because they're multinational and they have their hands in so many areas, and who can monitor, who can even try to say we're regulating Facebook. Nobody can say that. There's nothing even close to that.
Absolutely. They just got the largest fine that the British trade regulatory agency has ever given out, right? And part of that is that's all the British can do, right? Is fine them and hope that they pay, right? Like California is the only. I think you call it standing. I think in legal, like our government is the only one that can prosecute in this case, and especially when you're talking about multinational, international, transnational behavior. Yeah.
So there are a lot of stories in this article. It just goes on, and so if you want to find a topic, and again, even beyond these articles, there are a lot of topics that are interesting here, and touch on law and touch on other things. So definitely check that out. That's for later. So why don't we move on to there? Yeah, there's the link to Patagonia. I wanted to mention that Angela, and she was here earlier. She's going to be giving a talk on Wednesday on data trusts. And I think I mentioned this before. We've kind of gone through this talk, and I'll send out an invite to registration for a link soon, early this coming week. So Wednesday, and it's at 1 o'clock Eastern, which would be like 10 o'clock Pacific time, and then in Europe it would be in the evening, and in India it would be late at night, like about 10:30. And so if you can make that talk, please do. It's going to be It's going to be a nice talk. I've been working with her on the slides, so I think it'll be a good talk. And there will be, I hope, a robust audience and some conversation, and then it'll be recorded if you can't make the talk. So I'll make the recording available afterwards. But I think it would be nice to attend in person to support Angela. So please, when I'll post the poster in the Slack later this weekend, and we'll maybe try to make it to the talk. So let me share my screen again. And so next up is, I'm going to shift gears a little bit. Actually, let me go to the open chat forum. We stand up. I think this was from Saturday. There was an article on long-termism that is a philosophical movement, if you're interested in reading about that. I think this was more from last week where we were talking about this. Let's go to the VR channel.
Yeah, I saw that, and it's just funny because I saw a job posting from one of the institutes, Futures Predictions, Global Future. Anyway, it sounded like a really interesting job, but I wasn't familiar with the philosophy behind the institutes.
Right. Yeah. So in VR neurotech, there was something Jesse posted on Slava Bobrov, and Morgan said that he's an active member of Neurotech Accesses on Brain-Computer Interfaces. So it's an interview. This is an article on applying virtual reality to single-cell omics data. So for those of you who don't know what this is where you have single cells in a biological context, and they'll take single cells and they'll sequence and look at the expression of genes in single cells. And you get a bunch of data that, people usually will plot with some sort of method like UMAP or TSNE, which is a visualization technique for high-dimensional data sets. And they'll create these plots in 2 dimensions that are kind of like, almost like postmodern paintings. That, they don't, they have a lot of nice visualization value, but the interpretive value is somewhat limited. So what this group did was they created, they mapped the data set to a virtual world. And this is a person who's interacting with the data where it's in a 3-dimensional cube and they're playing with the data points. And so this is something that people have been doing for many years, visualizing data in these kind of environments. And, you can touch the data and manipulate it and move around inside the data cloud. So, the problem with this type of approach is that it's much like the 2-dimensional map of like a TSNE or a UMAP analysis, in that it's very pretty and it has a lot of visualization value. But the interpretation is still difficult because, if I can walk through the data, maybe and I can understand a little bit more about, where it is. But in real terms, it doesn't tell me the sort of, we were talking about networks before, it tells me maybe something about the global state or the global scale of the data, different relationships, like these 2 clusters, that things are, separable into clusters, or that these clusters are sort of next to one another in space. But what it doesn't tell me is maybe some of these points within the clusters, what are their relationship? Is there a causal relationship, or is there some other type, alternate set of relationships? So, this is just kind of like, they take the data, they plot it out into this VR environment, and then they visualize it. And so I think Morgan raised this point about, and we talked about this, the dimensionality reduction is really doing all the work. It's this method of dimensionality reduction. Because you start with data that's, maybe hundreds of dimensions potentially, and you reduce it to 3 dimensions here. And so that's what you're doing.
That's TS&E, right? I mean, that's its entire, it's how it was constructed. It's doing what it intended to do.
Right.
But I've seen this for so long in terms of protein folding and if we could just visualize this, and the visualization is not where the problem lies. It's grappling with the math and the physics, and eventually turning it over to an algorithm like AlphaFold. Right? So I mean, I to be positive, it's great for experimental control and stimulation, right? Like it allows us to do a much more naturalistic visual, auditory visual presentation for subjects or animals, and if you can be recording at the same time with that kind of naturalistic movement and interaction, like you get very different neural behavior, neural activity.
Yeah.
So much better than what we've been doing.
Yeah. Divyesh had to go. He had to go to dinner, so thank you for attending, Divyesh. All right, so we'll go to next. Let's go to Neuroscience Networks here. So there are a couple papers in here over the last week. There's a hackathon here, MICCAI hackathon. Is this like a...
Yeah, I didn't hear from, I think it was Krishna had a project about intensity normalization, maybe, or inhomogeneity correction. I wasn't sure. Anyway, MICCAI is like the big conference that is focused very much on, machine learning in kind of medical imaging. And so it does just a lot of different talks of people presenting their hackathon projects and maybe some of their research.
What time of the year does MICCAI happen?
So MICCAI happens in the fall. I don't know how things maybe got shifted due to COVID, but I want to say it's usually like September. Abstracts are coming up for ISMRM, which is another big, because that's all focused. It's MRI. A lot of good machine learning applied to MR data is presented there. Even though it's be a bit more applications oriented. I mean, you're going to see a lot of neural IPS and ICLR, et cetera, as well. But ISMRM is the newer, or the deadline coming, and then after that will be the human brain mapping deadline, which will be like January.
Okay.
Yeah. Speaking of like brain mapping, and this is a talk by Kate Jeffrey. This is someone who we covered last week in terms of spatial cognition, and this is on cognitive neuroscience and architecture. So this is like, this is from NeurotechX, and so you can check that video out.
Yeah, Neurotech Barcelona.
Okay, yeah.
It's a great group focused on architecture and neuroscience.
Yeah. And then, let's see. This is an article from Quanta. Brainburst can mimic famous AI learning strategy. So this is something that I found on... So this is where you have a new model of learning centered on bursts of neural activity that act as teaching signals, approximating backpropagation. And this is, talk about backprop. And then this is Blake Richards, who's actually one of the people on this paper. And so there's a problem called credit assignment. And that's kind of where they're coming from on this. They want to propose a solution to this problem of credit assignment. And where you're trying to trace neural activity to a certain set of neurons and understand learning a bit better in terms of what neurons are being activated and what neuron weights are increasing when. And so, ideally, if you're learning something about the world, you can trace a specific object to a specific set of activations, and that would be like basically being able to partition out a memory. So in psychology, there's this history of like the engram, where people think, well, that memories are stored as these specific things in the brain, and people, I guess, at some point thought it was like this little tiny area in the brain. We know now that there are networks that do this. But if you want to do this in a neural network, of course, you need to have this credit assignment where you can figure out which neurons in the model are being activated, and so that you can identify those, what weights are responsible for, say, identifying, say, the picture of a dog. And this helps with classification, because if you have a false positive, you can trace it back to sort of a sort of interpretation of the synaptic weights, and you can correct the model, and you can make the proper classification. So that's the value lies in that. It's a very nice line of work, and I would read more about that. Read the article and go to the source articles and read those. Next is, I was thinking a little bit, and I'm going to talk about the final version of the final version 1 of the Gibsonian information paper. So I was thinking about the fragmentation of representations during this. So this is a paper, and we talked about grid cells last week, where there are certain cells in the hippocampus that represent different places in the environment. It represents sort of the structural background of the environment. And then there's cells like head direction cells, which sort of give you sort of direction in the environment. So this is about grid cell maps. And so these are the cells that represent grids, or represent your space in front of you as a grid. And then your place cells and your head direction cells are all tuned to those locations. This is where they're looking at this representation, but there's a fragmentation of that representation. And so you don't think I'm kind of just pulling this out of my ears. This is something we were talking about with respect to this video, where they were building these things called Markov brains, and they were looking at like the representations that they built with these brains. They're not neural networks, but they're these sort of state machines that they've built that represent a brain and behavioral output. And they were looking at information flow versus information fragmentation. So when information fragmentation occurs, these networks are bifurcated, and so they're not, there's sort of a gap in between the representation of something and the representation is discontinuous. And I don't know what the consequence of that is, but in this paper, they had some rats running through a maze, and they were looking at how they're representing their spatial environment. And these maps were fragmented into repeating submaps. So, fragmentation can be good in the sense that it can break things down into subroutines, but it can also be bad because you don't necessarily have a unified representation. So this is an interesting thing that, this is an interesting area, and I don't know exactly where this fits in. Maybe this is more in the metabrains area, where we're dealing with different levels of representation. And so, when you have fragmentation, it can either be specialized sub-categorization, sub-functionalization, or it can just be something that just, blows up, and your representation is discontinuous, and it's actually a bad thing because you can't have a unified representation. So that's something I was thinking about this week, and I don't have a presentation on it, so it's not as clear as I'd like it to be, but in any case. And then Morgan put this paper in, Unified Treatment of Synchronization Patterns. And so this is
the Steve Strogatz.
Oh, okay.
Yeah,
Let's look at that.
and yeah, it really, yeah.
Yeah, it's actually has Adelson Mater on it too. Or this is, yeah.
Wait, my apologies.
Oh, okay, so that's fine.
Steve Strogatz is not on this paper.
Okay.
But Yunzhou Zhang is at Santa Fe Institute.
Okay, yeah. So Adelson's pretty good too. He's got a lot of interesting stuff to say about synchronization and unified treatment. Yeah, so this is about complex interconnected systems. One often has to go beyond the standard network description to account for generalized interactions. Here we establish a unified framework to simplify the stability analysis. So this is like dynamical systems and chaos. to simplify the stability analysis for cluster synchronization patterns for a wide range of networks, including hypergraphs, multi-layer networks, and temporal networks. So they do this, they look at this sort of framework. As an application, we can use simultaneous block diagonalization to unveil an intriguing type of chimera state that appear only in the presence of higher order interactions. So they're able to do this analysis of, they'll have what they'll have in a network typically is they'll build it from a connectivity matrix or a correlation matrix. And so you can do operations on this matrix that allow you to, find different things in that macro state that we were talking about. So they're able to do this. They discuss the method here, and they can apply it to a wide range of different systems. And they have these chimera states where when you get higher order interactions versus just regular global states, you get these chimera states of the network. So it's kind of an interesting paper. It's hard, it's hard to really get an appreciation for it unless you kind of dive into it. So I don't want to spend too much more time on it, just to say that if it's something that interests you, please follow up on it. We have the free energy principle. Yeah, Daniela actually is pretty, she's been working with some of the free energy principle people, and she's really interested in that. So if you're interested in the free energy principle, I would go talk to Daniela about some of that, because she's done a lot of writing on that, or she's done some research on it. She's done a lot of reading on it. So, so there's this paper, "The Emperor's New Markov Blankets," which is a kind of an interesting title. The Markov blanket is a concept in the free energy principle, and so it's kind of an interesting paper there. Let's see.
Yeah, I have asked Carl Friston about how how exactly I'm supposed to estimate that. In particular, when we're using high density EEG of say resting data.
Yeah.
And he's got papers that scale up to about 64 channels.
Okay.
And he talks about the difficulty in estimating these resting state dynamic causal models. This is to kind of operationalize it for us, in terms of what's a problem that we can get behind and this is data that's available. We've got this on thousands of subjects, 128 channel data. I still, it still seems to me like it's actually dimensionality reduction.
Yeah.
It's first projected onto the three top singular value components, and then.
Yeah.
But I'd love to hear more about that, because again in that particular paper where you're doing these coupled partial, you're solving these parameters for these coupled partial differential equations. Super important problem. Love to get at it, and if somebody's got work that would be relevant to that, I'd love to see it.
Yeah. Well, it looks like Ankit had to leave. He asked a question, spiking neural nets. I don't know what that meant. That was from a while back, about 8 minutes ago.
Yeah, I know Daniela doesn't. I don't know if it's because she doesn't have time or feel comfortable being on camera here, but I'd be really curious for her to just give a presentation about what's going on in the inference lab and what she's doing, and because I really don't know that much about it.
Yeah.
Is that the active inference lab?
Yes. Yeah,
because the regularity of their videos is awesome.
Yeah. Yeah, they really do have a good presence on the web and on YouTube. So.
And yeah, they've got a great. They had Ryan Smith talking about his kind of tutorial to active inference, and again, like great place to start.
Yeah. I think the spiking neural nets was in reference to the credit assignment problem. So that's what I think that was. And I guess, I don't know if they were referring specifically to spiking neural nets, but it was, I guess you could apply it to that and that would be something. So I don't know. So let's go back to this. Let me share my screen here. I'm going to finish up the Slack review and go to Bradenburg vehicles. I don't think there's anything pressing there. Just maybe a little bit here. This repository from last Saturday on evolutionary computation, and then Cognition Futures has a post on. This was something that Jesse posted on a convergent cross sorting for estimating dynamic coupling. This, I think, is some people that Jesse knows, and they posted this paper.
Yeah, I have the esteemed status of being Tony Shamaro's Facebook friend. So, who uses Facebook?
Yes.
Twitter follower. And Eric's the one from San Diego. He's a very interesting person who's like very pro cybernetics and pro like cognitive science guy. And it was looking like an interesting paper. I didn't get to look at it in any detail at this point, but it looked cool. And Tony liked it, so that made it particularly stand out to me.
It looks like a really interesting paper. Again, this is stuff with dynamical systems, so you have to have that background. And then I posted this. This was Jesse again. Genes, cognition, and human brain evolutions. There was a talk on this, and so. And then, so that's all for this week. So that's our Slack update. I know it took a long time, but I think that drives a lot of conversation. That's good. So, the next thing I want to talk about in our talk here, let's see. Kind of trying to figure out what to cover next. I think I will cover our. So let me talk about NeuroMatch. So the NeuroMatch submissions have been out, completed. We've, I think, got 2 or 3 from the group. Actually, I know that Krishna has one on his work, and I submitted with Ankit and Jesse the thing on switching. Jesse submitted the Gibsonian information abstract, which was with Daniela and Avery and Jesse and myself. And then close to the deadline, Jesse contacted me in the seller group with Avery, and they wanted to submit this thing on allostasis and allostatic regulation. So we worked on that, and I guess, did that get submitted, Jesse?
Yes, it did, and I appreciate you kind of coming at the last minute there. It's something that had been it sort of like finally hit all cylinders. Of the core of it is the core of Avery's stuff they've been dealing with for years and hasn't really had an outlet for. So that was part of the main thing for it. There was interest from 2 other people at Seller who are Jenny and Nick. And Jenny is let me start with Nick. Nick is very interested. I actually want, I kind of want both of them to tour the lab, honestly, like I do with all of my scholarly friends, apparently. But Nick, in particular, is interested in alternative or advances in educational things. So I think he really enjoys the spirit of the lab itself, what we're doing with open science and open source, and he's interested in a lot of educational ways of doing education. And I forget, I know he did stuff at, I forget if he's currently at George Mason or formerly at George Mason, but he's similar to me in my background of like politics a while ago, and then like data science-y kind of stuff, or going back to school for a technical degree, and he's familiar with agent-based modeling as well. So one of the components of the paper might be trying to lead into some agent-based modeling applications for what we're talking about as like an actual way to put either some code or just visualize things in a more simulation sense. So that's Nick, and Jenny is not really sure how to describe her, but she is very adjacent to Avery, and she has her own. I don't. Let me actually. I'm gonna, if you don't mind, share my screen.
Oh, go ahead. Yeah, please.
Ooh, will this work? Okay, Jenny is a designer and a creator.
Are you sharing your screen? Or?
I'm about to.
Okay. Good.
Getting the website up here. Here we go. And she made something called Plot Twisters, which is sort of it's a community of research designers and technologists creating opportunities for reflection, especially for young people. It's sort of a little bit of a Mr. Rogers neighborhood vibe, but much more formalized, and she's really interesting in working with technology and VR and AR, and it's sort of like, to be dramatic, like if Facebook is kind of like, the dark side of people trying to, come to the dark side and let's amplify your negative emotions. Jenny's sort of like, well, let's have some efforts that are in another direction about knowing yourself, and very Mr. Rogers vibe about it. This is obviously like very generalized terms, and but just she creates these sort of infrastructures for learning, and you can see Jenny here,
Okay.
and Nick here. And they're both currently in the UK right now. But they're an interesting group of people who are working on a lot of like modeling and helping development of inner world stuff, to be very general about it. So that's Jenny. And then I really don't have a good. Can I see this? Yeah, you can. I guess you can see this. This is sort of what we're working with. We had a lot of past iterations of the title, and I'm not quite satisfied with the title ultimately. But it originally started out as taste buds of relational qualia, and I threw out this like totally alternative one when we didn't really have a thing there about like cognitive agendas, and it kind of broke down into different ways of dealing with this. And I eventually kind of steered things into a context, content, conclusion. Thank you, Conrad Recording. I bet it did it because they're all like C or K sounds too. But anyway. I tried to frame things in a much broader biological sense for Neuromatch. The meat of what they were getting at is kind of about the middle part here, and there's I think this could be a little bit more crisp, but I think it's across the point of like we're trying to understand some basics of allostatic processes, and I figured like, all right, what can we actually turn this into a goal? And Avery's interested in this, in generalizing this across a lot of different domains, but also how I think another way to actually talk about this would be, we haven't used this word yet, but it came to my mind yet as a significant thing. It's sort of like almost like niches and how they affect allostatic points of stability for different either agents or different like how the agent is dealing with this representation of how they're relating to the subject matter, whether it's on the person or whatever else. So this is quite hand wavy right now, but it's been tough because Avery's health is in a precarious situation, and to be very, very general about that, and this sort of became a meshing point between the opportunity, and I brought it up, and it caught enough interest because I mentioned it before I mentioned it when Neuromatch first came out, and I was like, okay, yeah, maybe. But then there was enough sort of collective interest where we got it across the finish line. And I think that's actually really good because, you know, keeping this in the game and having something to develop, something to prepare for and present, I think that's really important for Avery and for everyone else involved as well in terms of keeping this kind of moving. So I think this is a good. There's a whole lot of refinement to do, and hopefully we get accepted. If we don't, we can still work on it. But I think getting accepted will be a little more impetus and pressure in a positive way for us to finish it out. But like at the same time, like really though, let me get this over here. I really think it will be once we all sit down, and I do think it'll be good for Jenny and Nick are important additions to this. Once we sit down and flesh out some things, like I tried to. My main influence right now has been a lot of organizational, administrative, and then putting the context and trying to get. How do we get? I realized like I'm outside of Bradley, of course. Like I'm kind of the most like okay. How do we functionalize this towards somewhat of an academic presentation? And it's sort of a being almost a diplomat between, yes, this is great research and stuff I'm interested in, and stuff I want to flesh out, versus essentially how do we get this to ultimately like a preprint kind of a status. That's kind of, it's been nice because Avery is totally on board with that, and even seeing Jenny and Nick also on board with that, I think that will be enough collective effort to get us there. And this is this has been on the this is something that's been on. This isn't just for people who aren't aware. Like this isn't just like, oh, we thought about this last week and all these ideas out of nowhere. Like no, this is actually like probably 2 or 3 or 4 years of thinking that's hopefully being crystallized into a preliminary presentation of sorts. So that's kind of where this is coming from, and hopefully it goes well, and we'll see what happens. But yes, and thank you, Bradley, for kind of coming at the end and offering some advanced editorial insight on how to communicate the kind of generalities we're getting at here.
Oh yeah, no problem. I think it's pretty good. I mean, it's a lot of problem with some of these philosophical types of abstracts is that you have all these ideas and it's like really deep, and there's so much to unpack, and you have to like condense it into this small space. Like, I want to mention this and this and this. So, part of it is like making it clear, kind of like the path from one end to the other. Like, what is it what's the core idea I want to communicate in the abstract? Now, with a preprint, you have more time to unpack things and explain. But like, using terms like, allostatic kinds.
Natural kinds, that was a whole like, oh, yeah.
And I mean, at Neuromatch, you're not going to have a philosophy audience, so they're not necessarily going to know what that means. But you can make, adjustments. You can say, well, we want to do this thing, allostatic kinds, is kind of like this. And then in the presentation, you would work it out a little bit. But also, and I think you've seen, like, I think at the Bika conference, there was like a philosopher there who did like, he tried to fit in his philosophy into 15 minutes. And it was like, but you notice that it was like an analytical framework. It was like, I'm going to approach a couple of examples and I'm going to walk through them a little bit. So there are ways to do this. I would, definitely, I hope this gets accepted. And if we do, then, the next step is, of course, the presentation, which is going to sort of unpack some of this. And then I would wait maybe until after the presentation is done to do like the preprint, which would be then the next step, and say, okay, now we got some feedback. We know kind of what works, what we can get out in a short, kind of compacted way, and then we'll sort of unpack these things in a preprint. And so it helps you not to like go all over the place, because there's that temptation to just say, oh yeah, it's this, and you're all over. And it helps you kind of focus down the argument to maybe a couple of examples, or maybe work out some definitions, and that's always powerful. I think it makes things more powerful in the end. Morgan posted this link, algebra.org.
Yeah, sorry, I wasn't sure exactly the direction this was going, but teaching, improving the teaching of STEM is certainly a goal, and just been thinking about this after going through a course on, what people are doing for coding classes for kind of STEM outreach. And this is a project that I want to follow up on in terms of how they're presenting math. And so
Okay.
I don't know if there's a subgroup for that kind of educational.
I don't know if we have an education channel. I think we have a math channel, which we might put some things in, and that might be.
I think there could be, honestly, because I've been wondering about that myself in some ways, and it's kind of adjacent. Not this whole discussion I have about the mentoring program that I'm envisioning, but not really here nor there at the moment. But no, thank you for sharing that. I actually will send that to Nick now, because he probably knows about it, or would be interested in it if he didn't. Sorry.
Yeah. Okay.
And just to follow up on what Bradley said before, yes to all of that, and also I think, as kind of been stated, I definitely won't be able to make a preprint of any kind until January.
Yeah, I know.
So, but I think it also is a good. How can I say this? I think it's very important for the continuity of some things that this happened. So I really wanted to make it happen when the opportunity came up. So that's great, and I take that as a major win. And more about that later. But outside of that, it's also continued experience there's the immediate stuff in the whole trajectory, the paper we're talking about, but also I think it's not a great time right now, but I think Jenny and or Nick I think they might have some really interesting things. Maybe I don't know if they're going to join a lab per se, but I think they're going to have some very interesting potential collaborative efforts ahead on various fronts. Particularly because Jenny is explicitly interested in some of the VR AR stuff that I want to get into. I don't know what crazy, but she's
here in San Francisco, yeah.
I'm sorry.
She's here in San Francisco, yeah?
I think that's their home base. I'm not really sure, though.
Okay.
But yeah.
We could meet at Noisebridge or something and talk VR AR for sure.
Okay, I'll keep all that in mind. But yeah, I think there's definitely going to be... I don't know how long they're going to be in the UK. I imagine for some more months, because they're there for a few months. The UK, the seller program, for US citizens, you have a 6-month limit. So it's sort of I don't know what their plans are. But I'll keep all this in mind. We can move on to
Yes, I know that limit well.
Thanks for the VR, gratuitous VR headset cameo. Yeah. Anyways, yeah.
I've got 3 of them lying around here.
Oh yeah. So yeah, we should do more practical stuff. I know we've been talking about VR and AR and trying to get more. And I've been trying to put together some resources for different, so the idea would be to get people who, want to do it on their end, wherever they are, to maybe have some standard content, or to have some, what kind of equipment do you need to buy? It should be fairly cheap. And this equipment is coming out now, so we can actually do some things.
This is a great way to tie in Neurotech X too, if anybody's interested. So I know that Neurotech X Paris meets regularly in VR with hubs.mozilla. Their kind of rooms, and yeah. And so you'd get a built-in kind of user base of people who are, certainly interested, if not developers.
Yeah, that's good. Well, we'll put a pin in that and come back to that at some point. I wanted to go through a bunch of stuff here. I have tabs open. And so the first thing I wanted to talk about was the Gibsonian information paper. So this is the sort of the final version of version 1 preprint. So it's been growing, and it's been getting more defined. This is the abstract that was submitted to Neuromatch. It's basically the same thing. Then there's some introduction. There's some more discussion about affordances, which was important to set up, to establish what was going on here. Talk about the sort of the nature of Gibsonian information versus Shannon information, which is very similar to, I think people are very familiar with that, and so contrasting the two, then going into the mathematical definitions, going into a diagram of what, so Gibsonian information is taking the external world, quantifying some of the information out in the environment, encoding it into a nervous system or an internal model, internal communication channel, and then decoding that into behaviors. So, you have, it's not really a brain, it's maybe a brain. In ecological psychology, there's not an emphasis. In fact, there's a de-emphasis on mental representations or neural representations. But this is something that, is part of the kind of built into this approach to information. That this is maybe some feedbacks, maybe some delays or displacements of information from the environment, but it's maybe we don't want to call it a representation per se. So that's sort of the idea. And then we have these examples of GI, I don't know, measures might be principles, but there's some terminology that needs to maybe be updated on this, but that's okay. And so we go through that. We go through some other things. 4-channel embodiment, which is this example of the rabbit where there are 4 sensory channels. There's another example from AR where you have these different sensory channels being engaged. And then finally, this is an agent capturing information on a set of sensors. It's a very crude diagram. And then, so that's there. And then the discussion. And so then we have, I think, up to 65 references here. So this is something I like to do. So this is something I want to post Monday. And I'm not sure whether it's going to go to bioRxiv or sciarchive. If you have, Jesse, if you have a preference, please let me know in the Slack. I think bioRxiv, it's more high profile, but sciarchive might be a little bit more appropriate. I don't really know. But in any case, when we get the preprint up, we can send a link to the Neuromatch people for Jesse, the abstract Jesse submitted, so we can submit that and they can have this preprint then linked to the abstract. And so one thing I like to do with papers when we prepare a preprint or publish a paper is we have this citation count graph. And so I showed this for the MetaBrain models preprint, but this is the one for the Gibsonian information paper. So this starts in 1949. There are a couple of citations in the 40s and the 60s and the 70s, and then some in the 80s to 91, and then and some in the 90s. And most of the citations are recent, the last 5 to 10 years. So that's here, and then that's here. So you see that we have this breakdown, and it's actually pretty good. I like this long tail of citation ages, because a lot of machine learning papers, deep learning papers, their profile is basically citing everything in the last 5 years. And that's something we try to avoid. I like this distribution quite a bit.
Yeah. That's cool to see.
Yeah. So next thing I wanted to talk about was the Metaverse. So this is the Wikipedia page on the Metaverse. So like I said, the Metaverse is an old idea. It's not something Zuckerberg came up with. The term arose in the early 90s. So like this is when VR, the first sort of round of really immersive VR started, but it was really clunky. And, this is where you get this hype cycle. So like, there was this hype cycle in the 90s of VR. You had like Lawnmower Man and The Matrix, and everyone thought it was like this thing that was going to, bust out. And it didn't bust out. It was the VR of the 90s was pretty clunky, but it was something new. And so it really captured people's imaginations. And I think ever since, like, I guess William Gibson, who's the science fiction writer, he really worked out some of these ideas, especially like in Omni magazine and in some of his books where, he worked out some of these ideas of cyberspace. And then that led to this metaverse idea where, you have this world that's in, it's a virtual world that you interact with people. So there are a lot of parts to this. They're massively multiplayer online games. There were things like Second Life, where you would have like real virtual communities of people who would just kind of come together. So all those parts were kind of being put in place. And now, we have this, people are trying to, I think, realize this vision, but it's a lot harder than we'd like because there are a lot of parts to it, and there's a lot of hype too, so it makes it harder to really break through. I think if we've talked about the hype cycle before in our meetings, and that is that, a lot of these technologies that capture the imagination and they really get blown out of proportion. And then there's this crash where people realize that maybe that's the case. And then, you start to, but at the same time, you start to get applications that are really kind of solid and that aren't hyped. And so you get this plateau of productivity. And I think we're kind of, with VR and AR, it's kind of like you have both at the same time. You have a lot of hype, but at the same time, you also have things that are productive. And it's kind of a weird thing. I've been looking for like a hype cycle for VR, and I haven't been able to find one. I know that this is put out by a marketing company, ironically, but I haven't been able to find a good, like we actually, I think Jesse and I have talked about this, where we're looking for a map of like this. So the summers and winters of AI, if you're familiar with that, where there's a summer of AI. And let me do my pad here. So like with AI, you had like a timeline that goes back to like the days of cybernetics, like 1950, and we go out to the modern era. And like we have these periods where we have like summers and winters. And I guess we're here now, or maybe a little bit above that. This is a summer. This is a winter. And the idea is that like you start, with AI, you start with these platforms that are, okay, come on. You start with these platforms that are, have a lot of promise. Like, you might start with a connection machine. And then the connection machine is, what was that?
It almost looks like a dinosaur, actually. So.
Yeah, no, this is not. I don't know why I can't draw here today. Let me write winter here. It's like the Loch Ness monster. Yeah. So the summers are like these technologies that come out, and people embrace them, and they start to play with them, and they get all these really cool results, and then people start to hype it and say this is going to be like this is going to go on forever, off into the sunset. And what happens is, like, when people realize the limitations of the technology, there's this winter. And so then there's this artificial correction where it doesn't get funded. So AI had a number of periods where it was funded very heavily, and then it wasn't funded. And then there was another innovation, and it was funded heavily, and then people kind of abandoned it, and so on and so forth. And I imagine that deep learning will have that same fate, although maybe not as much, because I think there's some maturity in that, when you have these peaks like this, there's some maturity over time that builds. So maybe this dampens a little bit. But, I mean, I'm looking for a map, and not as crudely drawn as this, but where you have, like, VR technologies like this could be the early 90s, where you have this peak and then the winter, maybe in the early 2000s, and then kind of coming up around the 2010s, and then down a little bit, and then up, and now the current era, and then maybe it'll go back down a bit. So we don't really know what. I'm looking for that kind of map, and I haven't created that map, and I don't know if it exists now, but that's something that would be interesting to look at.
Well, I have a kind of a related or adjacent question, and I imagine I haven't really seen. I don't know if I want like a study or what I'm looking for in this context, but I think the thing with VR and AR, which I'm in favor of, and I'm not anti those technologies, but one thing that I don't like a logistical shortcoming that I see is we're good at creating these sort of pocket dimensions, but for people, what people think the metaverse, I think like Ready Player One of these gigantic landscapes, and it's just one like the amount of market pressure towards individualization of content right now seems super averse to that. Or like, is there going to be a way where you can push specific like how would you even try to either have different filters for a large environment or very smaller environments? And I'm sure the people were talking about that, but it's like I find that to be it's like the when I think about at least, and I don't know if this is like just personally speaking, when I think of the metaverse in big quotes, and not even like Facebook's mumbo jumbo about it, but like I think a big virtual community, even I think this is like I'm not downplaying that either. Like I know, I don't know if I'm really saying I'm friends with him, but like Dan Fagella is like a writer about AI and stuff like that, and does their own emergy like AI consulting stuff, and they're pretty versed in where things are going. And they have a little bit of a pessimistic view that we're going to be kind of bottlenecked into, they have a concept called substrate reality, or the substrate monopoly, and how like who controls the substrate to the virtual world is going to have a lot of influence in the future. And like, okay, this is a little bit cyberpunky and negative or whatever, but like, I think there's something to that. I think there is going to be a push towards virtual life. But at the same time, like, I don't know how people are, outside of like, yes, Facebook is a gigantic infrastructure for tech stuff. Are they, I don't know, like, and I'm not even trying to say it has to be Facebook or not, but like, you see the dilemma here is there's so much individualization right now that's kind of shallow and ad hoc in particular. To have like a gigantic unifying metaverse feels incongruent with that.
Certainly, you see why. I mean, it's like a straight follow the money kind of.
Yeah.
It's like, why do you have that personalization, right?
Yeah.
It's because of advertising. I am curious about when you do simulations for multi-agent training, like how many people are looking at the kind of diversity of the multi-unit or multi-agent behavior? Because again, like the kind of procedurally generated worlds, you need, to get a kind of Ready Player One, right? You can't build these yourself, right? So computers have to build these worlds, but these worlds also need to interact, or they also need to be compatible, right? I think it's like a great case for again, like turning to simulations and seeing like what's our ability to try and create that diversity and maintain that compatibility, beyond StarCraft.
Yeah.
Which, again, is a really limited number. Or what are the good simulations for this kind of stuff?
I don't know. It seems like an open question, and So Jesse put "Hack the Planet," which is a reference to hackers, and yeah. So yeah, that actually leads me to my next thing, which is this. It's an update on NetLogo, and NetLogo is a
Oh yes.
it's a agent-based modeling platform, and this is not VR. I mean, I guess it could be if you wanted to, but this is more like simulating agents and figuring out what they're doing. There's a lot of heterogeneity here, so we want to like have specific problems like social problems or they're biological problems you can answer with this type of thing. Where you have a bunch of agents, you initialize them, you then have them interacting in an environment, and then they do things. It's a generative process, and then you can measure certain things like where are they in space, what kinds of resources are they interacting with. Basically network theory, but it's kind of like these agents interacting, and you can get these kind of global states. So this is the beginner's guide to NetLogo programming. It's their new version of their dictionary, and so they have these primitives that you can learn about. So this is, I think they've redone their dictionary here, where they have like the primitives, the linguistic primitives in the language that they're using to program. So you can run models off the shelf, or you can make your own models. And this is where you make your own models, and you have like the face, patch, scale, color, towards. These are just like calls that you can make. And then you have these tutorials, so you have actually models you can run. You don't have to code up anything. You can play with the parameters on them. And so you have epidemiological models, what they call the forest fire model, which is where you have patches of forest fire that kind of spread, and you can understand how those spreading events happen. And that's, of course, analogous with epidemiology. It's just a matter of like whether it's trees or humans or what you're dealing with. So this is definitely, agent-based modeling operates from that same place that network theory operates from, is that, you can sort of have these models that are very general. They could be neurons, it could be trees, it could be humans, it could be planets. They have similar behaviors, and if you put them into this simulation, you can understand the dynamics. And those are independent from maybe what's going on inside the object that you're modeling. Or, more specifically, you can specify those things in the model. So you can say that the human does these things versus what a tree does, and a tree does different things. And we can specify the behaviors in terms of that, but the dynamics of the interactions are the same or similar. So this gives you some information on how to start, how to make your simulation look nice, and different things. I like this. This is a nice educational tool. I know we've talked about NetLogo in the past, and we've used it in the past in the group. So definitely, if you're doing NetLogo type stuff, please put this. Maybe, I don't know, Jesse or Morgan can put this in the Slack.
Yeah, actually, I had a few questions about that.
Okay.
That's at Northwestern. It looks like that page was.
Yeah, they're based at Northwestern.
Is that NetLogo's basis, or is someone at Northwestern doing something separate to like? I know NetLogo's,
Yeah, they started at Northwestern.
They did.
There's a center at Northwestern where they do this.
Okay.
And they've made this public, it's made this very high profile, but it's an educational center, I believe, at Northwestern CCL, and they've been publishing it for years, so they've been publishing updates to it.
Yeah, but as you see from the about, it was founded at Tufts.
Okay, so they moved over. Yeah.
Yeah.
Okay.
Yeah, but yeah, I mean, I forget why I've seen this before, but it's been a long time. But has it received? I mean, on their initial page, I see I know some people don't update their years, but it says 2016 is the most. Okay, I see tweets from 2021. Okay, good.
Yeah,
good.
Yeah, it's
okay. Good.
I don't know how engaged they are. Yeah. On social media.
And I dropped in a link to Ski Run, not for the biomedical imaging, but I was trying to find their forest fire simulations because
okay.
Forest fire stuff. And do you remember the old Swarm package?
Yeah, I think so. Yeah.
Yeah, I'm only finding like really old stuff for wildfire, but it made me think of wildfire simulations. Yeah.
Yeah. I'm not sure that. Yeah, I'm not sure the simulations change that much, but yeah, if you run it on better computing, I guess it's. But yeah, I guess this is great. Yeah, thank you for this link too. So that's that, and we can follow up on that in coming meetings. I did want to go to this thing. I know we talked about this earlier in the Organoids channel, and this is something that I found yesterday. I was like just in the morning during breakfast, and I was just like. So I was just thinking about it, and so this is how Silicon Valley hatched a plan to turn blood into human eggs. And so it sounds kind of science fiction-y, but it's not really that science fiction. So apparently Silicon Valley is interested in funding startups that have a lot to do with fertility and reproduction. And you can see that there's a nice little graphic here with a stork and that. But this is the idea that they wanted to found this company to create an organoid that was an ovary. And so the idea was that you can create eggs from this organoid ovary and create humans from that. And so that sounds like a pretty audacious goal. And so, you probably have to know quite a bit of biology, but a lot of the people involved in this startup are like biologists by training. So it's a little bit touch and go in terms of what they're doing. But the goal is to sort of create this artificial egg. So you create an egg, you fertilize it with some sperm, and then you can have a human. And I don't know there are a lot of details in that I'm not going to get into right now, but suffice it to say it's a pretty audacious goal. So there's this company called Conception. So this is the company that they're working under. They're pursuing the official name for this is in vitro gametogenesis, which means that you're creating gametes or eggs or in a in vitro system or an artificial system. And so the idea is that you're creating this system. What they're doing is they're taking the induced pluripotent cells. So they're taking maybe like a skin cell or something, they turn it into an induced pluripotent cell. Then those induced pluripotent cells are turned into organoids. And the organoid then is, they're able to, there's been some groups that have done this where they've taken induced pluripotent cells and they've stimulated them with bits of rat ovary that they chop up they harvest a rat, they chop up these parts. And if you put the tissue in sort of the same culture conditions with this induced pluripotent cells, you can get those induced pluripotent cells to become that cell type. This happens a lot in mammalian development where you have signals that you have paracrine signaling and things like that guide the cell to a fate. And so, okay, Jesse has to go soon, but thank you for attending, Jesse. This is parthenogenesis, which is birth from non-fertilization. So they're parthenotes. So this is a form of asexual reproduction where the growth and development of embryos occur without fertilization by sperm. So this is something that you see in some modes of development in some organisms. There are some cells that are like this as well. But yeah, so that's a term that we want to learn as well for this. But basically that's the idea. They're taking they're trying to create eggs, they're trying to create these organoid ovaries, so they're creating them in culture conditions, and then those ovaries will produce eggs. And so there are a couple of drawbacks to this approach. It potentially would allow people at any age to have children, because you would just take a cell and you create an artificial ovary, and you could create an egg with your DNA. If it's a woman, it's easier than a man for certain reasons having to do with what they call exon activation. But if you can get around those barriers, then you can have an egg that has your DNA in it, and you can fertilize it. And you can, in fact, have multiple DNA from multiple donors in it. And so there are a lot of ethical issues with this. But that's the upshot. The downshot, of course, is that you don't have what they call a germline, which is that there's no germline, so it's not protected from somatic mutations. So your cells have a lot of somatic mutations that they accumulate over your life. And those things can, turn the cell cancerous. As you age, you get more and more of them. That's why older people have cancers, like prostate cancers and other types of cancers. Those that isn't repaired when you rejuvenate the cell using induced pluripotency. So those mutations are all still there. So the germline is where the genome is protected in germ cells right at like embryogenesis, and you don't have a lot of these somatic mutations. So that's another thing that's a problem. But there are some groups in Japan that were doing this kind of work where they were converting iPS cells into and making these ovary organoids, and so that's the way that they're doing this. So they've been trying to do this for a while. They're very enthusiastic, and maybe they'll be able to pull off something. It's not clear to me also that, like, if you're taking a pluripotent cell and you're creating an artificial ovary, if that ovary is going to produce a totipotent egg, or if the egg is going to be pluripotent. I'm not really sure. It may be that, like, it does what an embryo or what a ovary does, and that's what it does. But there are a lot of technical questions, is what I'm saying. So there are a lot of ethical issues also with using fetal tissue. So do you want to use tissue from an aborted fetus, or do you want to use is that something that's not something you want to do? So there are a lot of, like, kind of source donation issues of source donors that are a barrier to this as well. But this is the paper one of the papers that they cite in this article, and this is a science paper from 2021 actually. So this is a Japanese group, and they've done this stuff. They've generated ovarian follicles from mouse pluripotent stem cells. So this abstract reads, oocytes mature in a specialized fluid-filled sac, the ovarian follicle, which provides signals needed for meiosis and germ cell growth. So this is where you're creating these conditions for oocytes to mature. So what happens in an ovary is the ovary releases an egg, and then it implants in the wall of the uterus, and it's able to grow. And of course, if it doesn't implant, then you have a problem, because it actually, there's another method that's much older than this, and it's indirect reprogramming of cells, where you take a somatic cell's nucleus and you put it into an egg and you implant it into, say, like, they've done this in farm animals like sheep or in goats or in cows, and they're able to take these eggs that they put the somatic nucleus into and implant them into their ovaries. And if the cells implant, then they produce basically a clonal organism, which has the same DNA as the parent. And so this has been done kind of like in that way before, but this is where they actually want to create the reproductive, sort of an organoid of the reproductive organs, and then create these conditions where you can get a gamete to, be fertilized and mature and then produce offspring. So they've been doing this. Work, and this term niche, which Jesse mentioned before, this is also something that you find a lot in this field of, like, stem cell biology and organoids, and that is that you have to create a niche. You create conditions in the cellular environment that are conducive to differentiation and growth. So, a lot of times you can do all these genetic engineering things, but to really make something successful, you have to create the right niche for it. And that's creating, like, the right culture conditions, the right type of, like, conditions biologically, being near cells of a similar type. So all this, and that's kind of the magic. There's this niche that has to be created, and that's where the magic is in that kind of approach, is that, how do you create those conditions that allow for these things to happen? It's not enough just to have the biological hardware, so to speak, like the gene expression, the right tissue type, like if you create an organoid, but also the niche conditions. Is it in the right location? Is it in the right media conditions? That sort of thing. So that's...
Yeah, and it entails, understanding the complex behavior. I mean, the complex system, right? I mean, this is why, somebody, I was at a, birthday party for friend's daughter's friend, and this ecologist kind of apologizing for being ecologist. I was like, hey, you guys brought complex systems to the forefront. Stealing lots of things from ecology. What are you talking about?
That's true. Now, ecology has brought us like complex systems, also network theory, kind of like interactions, looking at and mathematically too, not just like
right. The binomial mixture model stuff that I did my PhD work on was actually like all the papers on it were all from ecology.
Yeah.
It's good stuff.
Yeah. So I think that's it for today. I know Jesse has to leave, and well, thank you for attending, and hopefully we can, maybe if we can invite some of the people that we talked about today who want to join the lab. Maybe, I know we've been doing some recruiting where we've been putting out calls for projects and people have been responding to that. But also people are just interested in doing things that of their interests, and it kind of matches up with what we're doing in the lab. Please, let's reach out to those people, or they can contact us and we can maybe do something as well, because I don't want to constrain people to any one thing. This is kind of the one feature of this lab, is that it allows you to kind of take ownership of things. And so if it's something that they want to do, that's not exactly how we describe it, we want to encourage that. So
yeah, we have to follow up on some of the applications, and I haven't really had any time or capacity to do that. There's a whole, I think that's going to be something, I think that that'll be a big thing for the winter. I think will be kind of going forward with that, because there's a lot of stuff that could be done in the lab. And as far just like big picture sense or future sense, as far as Nick and Jenny, absolutely. I think, and I wouldn't be surprised if in the next couple of weeks, especially because of the NeuroMatch formatting, like even just having, it may be during the meeting here, or just it might be nice to schedule a group call with all of us, Bradley and Avery and myself and them on the paper, or on the presentation for NeuroMatch. So if you have time, if you have a specific time, they're obviously in the UK, so the time block's later, but like this, they have flexibility in that. So maybe we could, I'll be in touch in Slack about that, because I think that might be helpful for everybody. But that's all I have to say, and it's good to see everybody, and take care.
Yeah, take care. And Morgan, I know we talked about, like, you had asked for advice about moving forward on the organoid stuff. So
yeah,
I didn't know kind of what you wanted in terms of advice, or if there was like something that we wanted to follow up on, or.
Well, I mean, I'm still. I mean, it's early for me in terms of doing the kind of background research and gathering lab information and things to see, like, but yeah, I would definitely take any leads in terms of labs that would be interested in sharing their protocols. I mean, first thing I need to do is really talk to the brain engineers group at Santa Cruz, because it seems like if any group is set up for that, it seems like that's exactly what their papers are about. Yeah. But also getting someone who's doing this now and willing to talk about it, hopefully, like, with us, here, would be great too, just in terms of, somebody who can compare, I mean, there's definitely some companies that have a protocol, like, will sell you organoids, how does that differ from the kinds of things that Alison Moitry's doing? Is it, I think it's Kriegstein. I forget there's the director here. I'm pointing over my shoulder because UCSF is up the hill here.
Okay.
Or I should say the Stem Cell Center is up the hill here in particular. Is Berkeley, what labs at Berkeley are using not stem cells, but organoids, brain organoids, right? Are there, how about Davis? Other places, the paper that I've pointed, certain like synthetic bio people to, in terms of can we synthesize this growth factor, at like order of magnitude less cost that is essential for cerebral organoid growth. That was from a Northwestern group, so I assume they are also, I assume they're using their own technology, and then, how to compare the kind of the opportunities afforded by the different kind of bio incubators, right? So it seems like everyone has in life sciences is getting their own, every university is getting its own life science incubator. And how useful would that be? In this case, like organoids are definitely going to take a lot more infrastructure than the kind of labs that have been set up to like GenSpace and I forget the name of the biolab in Boston, but the reason I dropped Tim Stiles' name into the cerebral organoids, brain organoids channel was that he's from that group in Boston. In addition to making software that's absolutely relevant for this. This definitely takes infrastructure. And so, but I'd really like to find a place where, we can kind of trying to keep to your position paper or, the About Us paper, how lean can we do this? Right. And we need, and another reason, and this gets to, why Moitri's work in particular is so important, is that they're growing they're aging those organoids for really long periods of time. Is that's what we want to do too, right? That like there's all sorts of great science to do from that, and that means like not only infrastructure but kind of like maintained infrastructure. This is more than anything is something where it's like it's not just grow it and then you have it, and this will be an ongoing thing, right? And hopefully we'll just keep the pipeline going in terms of, there'll be older ones and we'll keep putting in new ones that will, have different changes, right? And that's leaving aside the kind of necessary imaging work that I think the Silva paper gets at, right? Is like, so you've grown a brain organoid. Are you tracking all of its cells in 3D, right? Like what kind of single cell information are you able to get from that organoid without destroying it, right? And because potentially there's going to be real bifurcations in the genetic expression at some point in developmental time, right? I mean, this is why it's the holy grail of developmental neurobio, right? Is that we get to grow and modify and experiment on a quote simple system.
Yeah.
But we want it to approach real life complexity, right? And so track it, like there's a separate second track of technology that is the 3D tracking and kind of like, you know, cell informatics that you need to be doing for like that organoid. And, you know, again, like hopefully you've got, you know, thousands of these. Yeah. So again, that there's a question, you know, that infrastructure hopefully could again be, you know, kind of rented for time in the sense that like you wouldn't have to necessarily have that, or I don't know, you know, like I'd love to learn what labs are doing here, and I feel like that's definitely going to be like a smaller number of labs that are even trying to do that. You know, I think there's a big like let's grow these and then destroy them in some search for omic differences, right? Which, you know, again, like this is why the graph theoretic criticism from John about, you know, like it seems like you guys just use graph theory measures to separate, you know, separate groups. It's like, oh yeah, we have no idea what we're looking for.
Yeah.
So that as well as, you know, it would also be nice to get a sense of like how you know, how feasible is this, you know, now, or should I, you know, like there's definitely a big draw to go back to working on, you know, neuroimaging related projects, because that's my experience. And, you know, so like a big, I have a big question in my own mind about what kind of retraining is really necessary to you know, start to claim to be, you know, to have that as be my like research focus, you know, and more so than like probably a lot of people, you know, as someone who's in like analysis or methods group, you know, I was the only one with a molecular biology background.
Yeah.
So, you know, like I've got a bit of a head start, or, you know, like I feel comfortable talking and reading these papers, but I know the difference between reading a paper and doing protocol, and especially doing protocol at scale.
Yeah.
Right. Yeah. Like all sorts of differences. And, so, you know, I see some opportunities at say UCSF where, you know, I could be in a lab that's doing this kind, you know, not, let's just call it IPS, you know, kind of stuff, right? Like, you know, how much should I do that first before jumping to, you know, growing Daleks, which is, you know, of course, the end goal here.
I think, it'd have to get started at some point. I don't know how much retraining is required, because it's like it's a special skill even within, like, you know, stem cell biology. It's like, but I think the first step would be to just get like an accounting of what people are doing and maybe the opportunity, the landscape, basically. And then we can, because I've talked about this to a couple people, and it's like we have to get a good starting point, and we haven't really kind of figured out where that is. Like doing it on your own maybe is not the thing. You know, maybe even just a collaboration too is good.
Yeah, but you know, one of the reasons that I dropped that, I forget what their name is here. Let me just remind. Conception Bio, right? So it's pretty obvious from Conception Bio's own words, right, that this isn't like the people who got this started aren't necessarily the domain experts.
Right. Yeah.
Right. And I want to recognize that as well.
Right.
You know, that how important is that for, like assuming that we're doing assuming we're doing a startup with a benefit corporation focus, right? In the sense that like the brains are grown, the data is made available, or like the public benefit is a part of the corporation's goals, right? Yeah. How important is it that the founders actually know the molecular biology protocols, or things like that? Because certainly again with the organoids, it seems like you're obviously talking about a team. I mean, this is what I get from the Tarasky talk from David Mooney, right? Is that they're bringing together on purpose, biomaterials research, like knowledge of, Navier-Stokes, there's like physics, there's, I still think about the great group that presented at IGI, where it's like, they're doing, CRISPR, and they're all laser physicists, right? It doesn't mean that that you don't have a domain expertise. Right. It just isn't necessarily in that particular, in kind of like that startup vehicle's focus. Or it's just one of the things. But I absolutely agree. Certainly my thoughts is that like I have to do the survey of labs, survey of startups and other companies, because again, like I definitely assume that big corporations have their own internal research efforts that we don't hear about necessarily. But it would be crazy to not be looking at some of these first aid drug testing neuro drugs, right? Because as you know from The Inflamed Mind, great book about changes in psychiatry, especially from the drug companies. Drug companies got out of psychiatry a number of years ago because, hey, like how do we do this? And one of the VPs said to him, it's like, the only way we're getting back into psychiatry is if there's like a major change in how we do the drug development, right? I think organoids would be certainly one possibility where that could be the kind of change that they need. Anyway, putting trying to get some of those threads, and then there's actually some really nice issues in organoids and what we talked about, like what are the major arguments in the field between labs, or what are the labs trying to work out between themselves in terms of what's the best approach, right? And yes, and I will still have that video from UCSF as the prime example of but so anything on those, I'm still looking, and still would love others opinions on that, and
I'll if once you get something together, why don't you let us review it in the Slack, and it may just be me, but we can get a conversation going. I think once we get like some sort of what the landscape is, and we can say how do we exactly want to do this. We'd even been talking about secondary data before, where different labs will publish their data, and that may or may not be useful, but that's also something that we want to keep in mind for depending on the questions we want to ask. And then after we get the this overview, then we can go on to like, okay, how do we want to approach this? Then we can say like, what's the gap here? Is there like a gap that people aren't addressing? What are our resources? Because our resources are limited in some ways, and very rich in other ways. So how do we leverage our strengths on to what they're doing or what people are doing? Maybe it doesn't make sense to create a huge wet lab enterprise, but maybe it does make sense to say, like, do some smaller piece that's, no one's doing it. So.
Yes.
All right. Well, thanks for meeting. And we can talk more on Slack, or if you want to meet like another time, we can set up a meeting as well. So. All right.
Thanks so much. Again, amazing links and amazing food for thought that is on Slack there.
Yeah. Okay. Have a good week. Talk to you later. Bye.